to do today, and I'd like to have your full attention. I'd also like to have, I'd like to be a science student. You're a second year science student, and all I'm asking you to do is just to try to be interested and ask questions. That's what I'd like you to do, because there's a lot of things to learn, and what I don't know is, what are you doing at home? I mean, normally if you're taking an advanced class, you wouldn't just come to class. You'd do stuff at home, and you do stuff in class. And so you want to make sure you're not just doing stuff in class, all right? So I want to show you, the first thing I want to do is, you can take a look at this right here and see how well you thought you did. Oh, Jeff's not done, I'm sorry.
Sorry,
Oh, we gave you too much.
Do you have quizzes?
Yeah, I have those. Okay. Did everybody took the last quiz on octane, right?
Yeah.
Yeah, never.
Okay. All right. I'll give it back to you in a minute.
Wait, I don't think Jeffrey has.
That was your thing.
I think he did. Yeah, he did. Wait. If you take a look, I don't know what your 5 favorites were. This side over here. Hang on. How do you know this side would be polar? What are you looking for when you look for a polar amino acid?
OH.
That's more likely. And there's another character who unevenly bonds with hydrogen. He's nitrogen, right? So when you find nitrogen hydrogen or oxygen hydrogen, then that's going to be a polar. So this side here is all the polar ones. I don't know which one you picked, but you get pick your 5 favorite ones. I thought this was kind of hard. That's why I put it as extra credit. Somebody just liked the challenge of an extra credit, so I put that on there. And then on the other side, you'll notice that the R groups, here's a plain old hydrogen, here's CH3. There's no real polarity there, so these are called the non-polar ones. All right. The reason I also want to show you is that why were we doing this? I'm going to uncover this. So we had this the other day. The last part of this chapter talks about biological molecules. Now, of those, one of them is not a polymer. Okay, so let's take a look at the first one again. Proteins, absolutely one of the most important things a cell can make. Even the Proteus means of first importance. So the most important thing a cell makes is its proteins. So proteins are polymers, but what are the amino acids that put them together? What are the monomers that put them together? Amino acids. Now we know there's millions of proteins. Some maybe 80 amino acids long, or 503, or 265 long, and there's so many of them, and they have a different sequence of amino acids. And we know that it said here, if you remember, I got this out of an AP biology book. I'll show you what I have here. What is the primary structure of an amino acid? What's the primary structure? What's the most basic level of a protein? Yeah, but it's the sequence of amino acids, all right? And so the sequence of amino acids, just like this pop bead thing I had, the sequence is the primary structure. And then the book talked about a secondary structure, and they only talked about 2 things. There were 2 things that you have all these polar and non-polar ends sticking out, and they can attract each other or repel each other, and they can actually twist themselves into, what's a secondary structure? What's another name for a spiral? A helix. And they can also make structures that are kind of like accordion, bent back and forth, and that's called a pleated sheet. And those are called secondary structures. So you got the primary structure, secondary structure, and then what you'll see sometimes it will fold itself. Here's a helix right here, and here's a pleated sheet. But it might fold itself even more and turn into a tertiary structure like that. Now, I usually don't talk too much about quaternary, because that means you might have, here's a protein here, here's another protein, here's another, but at least know 1, 2, and 3 tertiary there. Here, they show an example out of the biology book of why this part might be attracted over here, and it might draw it into a little loopy thing right there. And I showed one of these the other day in the book. This might be the final, what the protein looks like. And I still think it's amazing that the sequence of amino acids have to be right, the folding has to be right, and then you have this shape, and it acts like an enzyme, for example. And everything has to go right for the enzyme to work, and it works all the time. in my cells, the enzymes work all the time. They're working, and they work right. But I also tell you that these are very sensitive to changes. Your book talked about, what's the vocabulary word that means that you've either changed the pH or the temperature, which caused the protein to become not its normal shape, and it doesn't work anymore. Do anybody know that fancy word? Denatured. And that's why, remember the word buffer? Before I started teaching, I actually worked in a lab, and we had to extract some proteins, some enzymes from cells. And so you actually break up the cell membranes, and you spin them down in a centrifuge, and you find the part that has the proteins of the enzymes. And we had to put it in a buffer solution. Why? Also, I always had it on ice. Always had ice buckets there, crushed ice. I put them in there because I couldn't let it come all the way up to room temperature, so I had to kind of keep it cool so it wouldn't denature. It had to be in a buffer solution, so any possible change in pH wouldn't change the shape of the thing. And so if you're doing enzyme studies, it costs a little bit more money to do those, but those are essential. So if you ever go to college and they ask you to work in a lab and you say, we're going to do some enzyme studies, you'll probably have a lot of ice, you'll have a centrifuge, and you'll probably have buffer solutions like that. So I want to show that to you. And then, so we're still on protein here, and I pretty much finished that, but I'm going to show you one other thing to talk about in the book. And I have two little models here for you. Oh, can you go over there and give me, I need 6 white hydrogens. 6 white hydrogens. I forgot to get those out. So I'm going to show you something that at one time, it's about 16 years ago when I first started teaching this, they had a word, and the word I believe is on page, I think it's on page 1032. And it's a word, when I saw it, I had never heard it before. And of course, I probably thought at first, well, maybe it doesn't mean very much because I hadn't heard it before. Then I started looking into it, and it was way, way more important than I thought. So I want to make sure that you've heard this word. It's a really big chemistry word. You can impress a lot of people if you tell them about this word, okay? So here we go. Everybody see these two? Okay. Now, if I ask you, are there any isomers of methane besides that one? Say, I can't think of any other arrangements. So what do you think? So I try to make it, it's the same thing, isn't it?
Yes.
Okay, so let me show you what I'm going to do now. I'm going to replace a hydrogen with something else. Okay, now, are there any isomers of that besides that one?
No.
What if I put it over here?
No.
Now, they're actually, can I superimpose that?
No.
Yeah, they're identical. Superimpose me there. So, okay, no problem here. Let's try another one.
Yeah.
Let's replace another one.
Oh yeah.
Are there
No
any
isomers.
is
No
that
isomers.
any isomers besides that one?
Only one.
No,
Only
no.
one.
No.
No isomers.
Oh,
Okay, let's take a look.
there. No.
Can't superimpose it.
Can we or not?
Can.
Yes, you can.
You can.
Yeah.
Obviously, you can.
No, you can't.
Yeah, right there.
Only one isomer. One.
Okay, all right, hold it.
Down there.
Wait, I'm getting to the good stuff.
There.
Okay, ready?
There.
Now, so what happened is they found out, and this is kind of interesting.
Bet with 3 it's different.
It was kind of 4 actually.
Oh,
4. I
that's a
need
yes.
one red one. Sorry, one red one.
Oh.
Get to work.
Go. Mason, you're not going fast enough.
Shh, 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 shh. Don't do it. Don't.
We just told you. Hey! Opa!
Okay, now let me show you something. All right.
Mason's just like.
So what I have here is it's like a Rubik's cube. You say, well, look, take a look at this right here. All right, what do you think about that?
Nope.
Superimposable.
Yes.
Okay. What do you think?
Yes.
Okay. So, all right, what I'm going to show you is now. Let's change one more. Wait a minute.
Whoa.
Wait. I'm sorry.
Hold up. What you doing there?
Wait a minute.
What's going on out there?
Sorry.
That's good.
Oh. Making big changes.
Maybe we don't.
What?
That's
All right.
superimposable.
My goodness.
Okay, let's try it.
Oh. Oh.
Oh.
Oh.
Okay, so what we have here is kind of a weird thing. There is actually another isomer of this one, but you wouldn't say structural. It's not a structural isomer because we have the same atoms, don't we? And it's called an optical isomer. An optical isomer means it's like a mirror image. And they have this word called chirality, and chirality comes from a Greek word which means handedness. It means glove, like glove, okay? Anyway, right here, I can put a mirror here. This is a mirror image, but I can't superimpose it, so it is different.
Oh, because it's.
And for a while, they probably thought they didn't understand, well, that's not really an isomer, and it's not going to cause any difference. But it did. And so let me show you what happened here. If you'll turn your this page here over. It says chirality. Why is it important? And this is called optical isomerism. Right now, the only thing you know about is structural isomers. These are called optical isomers. Molecules said to be chiral if it can exist is isomers called enantiomers, and they are non-superimposable mirror images of each other. We often say these molecules exhibit handedness after the non-superimposable mirror image left and right hands. These things rotate polarized light. I've used that once in college. I actually had a time where radar light goes in every direction. Light goes in, like a light bulb's going off, and light's going this way, and it's going in every direction. You can get what they call polarized light, and what it does, it kind of, all the light that's going this way, all right, for example, let's say if light's going like this, it'll get blocked. If it's going like this, it can get through. So you can use a polarizing filter. I have one here. You can use a polarizing filter that only lets light go in one direction. Now, yes, I did get this at a 3D movie, and yes, they did want you to put these in the bin when you got done.
I didn't know you were supposed
But-
to put them in the bin.
That you're supposed to, but they know
I took a couple.
that not everybody does that. So
Wait, what do you do?
So just,
Wait, I'm-
just because other people are doing
Do you
it,
use some 3D glasses?
okay, I'm trying to show you how this works. Okay, now anyway, these are polarizing filters, and this filter, whether you know it or not, one of these lenses is polarizing so that you watch a movie that the light comes this way. And this one's 90-degree angles, and you get to watch a movie, it comes like this. And so they actually had two movie cameras shooting at the same time. And one of them was over here, one over here. And that's how you get three-dimensionality. Because this eye sees a slightly different picture of my finger. This eye can see a little bit further on this side, and this eye, and that's what gives you three-dimensionality. So they actually film it with two different cameras, and then they want your brain, they want this eye to only see this movie, and they want the other eye to see the other movie, and then it looks like three-dimensional. And so how they do it is they use polarizing filters. Now I'll show you how these are polarizing filters. If I use this in science, I could shine light through here, and they actually make rocks that are really, really thin. You can do it with Scotch tape too, but you can shine that light in there. It'll give off different colors. And you can actually identify different rocks through polarizing light. Okay. Anyway, through polarizing light. And I want to show you how this works here. If you have polarized sunglasses, and a lot of you have sunglasses, that if they cost more than $5, even the $5 ones, some of them are polarizing lenses, all right? If you have a broken pair, and you don't want them anymore, but what you can do is kind of neat. You can tell whether or not they're polarizing lenses. And so what you do is, if I put them like this, you can still see through the light, can't you? Light still comes through them, right?
Yeah.
Now what's what happens when I turn it at 90 degrees? And a scientist would have two polarizing filters, and they would rotate them. Rotate, rotate. Okay. So now I'm blocking light from both directions, and you can always tell if your sunglasses are polarized, if they really are polarizing sunglasses.
I'd say those are pretty polarized.
Right, they are. Now, anyway, you can also rotate it, and what sometimes science will do, they'll rotate at different angles to see what happens to the light, and that's how they can identify things. So let's read on this article. They didn't think this meant much for a while, but here's the first time it happens. Look at amino acids. Amino acids, if this is not hydrogen, if this is not hydrogen, then you have 4 different things attached to that carbon, don't you?
Yep.
That means it can be chiral. That means you can make an optical isomer like I did over here, right? So when you have a carbon attached to 4 different things, so glycine would be the only one would not have an optical isomer. Okay? Now, so let's read on here and say, so what? So what? Therefore, in designing pharmaceuticals, we actually make medicines. And sometimes the medicines are organic, and sometimes the carbon is attached to 4 different things. Well, so what? Okay, so here's what happens. It says sometimes one of the optical isomers, it is the one that makes you better. And the other one, which is a mirror image, sometimes it doesn't do anything, and you're okay. But sometimes the other one hurts you. And so what they thought was a medicine, if you have too many of the other one, it can hurt you. So here's what companies did, all right? And I'll show you what I was talking about here. I'm not your biology teacher, but I want you to imagine this, all right? So here's a chemical that's supposed to land at a drug binding site, a place that on the cell where it's supposed to attach and it's supposed to do its thing. And it's supposed to have A here, B here, and C here. But what if you have the other optical isomer, then you would not be able to match A, B, and C. And so this would be ineffective, and this would be the one that's effective, okay? So that's what they're talking about by being ineffective. So here, I'll show you a fancy word, and you'll seem very smart if you say this. How do we make drugs then? If we want to make drugs and only have one type, it just so happens that living organisms only make one of these. But when we make it in a chemical laboratory, artificially, synthetically, it makes even amounts, about 50% of each. So we can make them a lot cheaper in a laboratory than wait until living organisms make the drug and you have to extract it from them. But, so here's what happens. Look at this. Some companies, even today, they will give you what they call a racemic mixture. Now, racemic mixture means it's got the active form in it and the inactive form in it, and they say, you know what? Well, so far, we don't think the other part hurts you, so we're just going to give it to you, and only half of the medicine they're giving you actually is effective. Now, sometimes, though, if you make a racemic mixture, half of that might hurt you. And then, so companies are now starting to go back on all the drugs they're making to see which ones are chiral. And now how are you going to separate them? One way is that you can actually one of these optical isomers will rotate light at, let's say, 60 degrees, and the other one will rotate 60 degrees to the left, 60 degrees to the right. So we could identify which one it is, and then you have to separate, which is expensive. It's expensive to separate optical isomers. Yes.
Are the companies being forced to do that, or are they just going back and doing on that?
At first, nobody was forcing them because they didn't have any research showing that the racemic mixture was hurting anybody. But now they're finding more and more cases where some people having that inactive one is hurting them. So it says right here, that's a great question you had, right? It says here, however, it is a procedure that is growingly increasing controversial to do the half and half. Evidence mounts about the inactive form may actually produce detrimental effects. So companies are having to spend a lot of money. They're having to go back. They don't want to be sued either. They're going back and spending money and trying to figure out every drug we've ever made, is it chiral or not chiral? We might have to start making it differently. Anyway, if you ever do go to college and you do something like this and you hear about optical isomers, you may hear them use the word right-handed. That means polarizing lens tends to be deflected to the right, or sinistral. I'm sorry that word, sinistral. It came from a word means left, or I know you don't like it. The word left-handed means sinistral, like sinister, and I hate that.
Sinister?
What?
No. Because the word sinister,
Sinister,
in everyday language, the word sinister means evil. At one time, people actually thought left-handed people, there was something. You're talking a couple thousand years ago. All right, question, real question.
Master
Yeah, quick question. All right, quick question. I gotta go. Quick question on that. Anyway, I thought that's pretty neat. That's pretty neat stuff. Yes.
So what are like some of the side effects that are common when you're taking these things?
Honestly, I would have to look it up because I would give you credit if you want to look up, say, inactive optical isomers, detrimental effects, or something like that. How many drugs are there out there? I don't know. How many are chiral? I don't know that. But if I had a drug company and I made 20 drugs, I'd be studying all 20 of those drugs to see if they are. Yes.
Could you explain what about these chiral drugs or certain chiral isotopes of drugs makes them
Isomers.
dangerous?
What are they? Isomer.
What makes those chiral isomers dangerous? Like, is it
Okay.
they bond?
That's a good question. The fact that they don't bind to the place in the cell we want you to, they're still floating around the blood. So now, do they go and affect some other cell? Do they? And that's what you're going to look up. What are some of the detrimental effects? I don't know that offhand.
It's the fact that they don't 100%.
But we're getting more and more evidence showing that, hey, we're being hurting people. We didn't know that. We were hurting people because we didn't want to separate them.
What was that word you used? Chiral?
Yeah, right. And if you look right here, I'm on page 1022. Here's what you're going to see on the test. The test is going to ask you which carbon is chiral. Okay, and I'll show you how the first example they have here. Now take a look at this carbon here. First of all, he has to be attached to 4 different things. Okay, now watch. See this carbon? He's attached to that and that. They're different. And he's attached to this big chunk thing down here, right? So he can't be a chiral carbon because he's only attached to 3 different things. Let's go to the next carbon. This carbon is attached to a COH, OH. Are all 4 of those things, and this right here, is all 4 of those things different? Yes. So that's a chiral carbon. What about the next one? This carbon is attached to this big chunk, this OH, this H, and this chunk down here. And so it's chiral because it's attached to 4 different things. Now down here at the bottom, it's attached to this, and this big chunk. So I circled the 3 chiral carbons. But if you were to take your book home, they actually did a good job actually putting squares around the 4 different things that you see. And so anyway, that's what's called a chirality. All right, so anytime carbon is attached to 4 different things, there is a possibility of having an optical isomer. Okay, everybody okay on that? All right, so I'm done on that. What I'd like to do now is let's go back to our original outline. Now, not only have we talked about proteins being a polymer and how they come together, matter of fact, remember how we talked about the H from one of the groups as bonded with an OH to another? Let me review that.
Have there been any positive effects of chiral?
Well, I don't know. I have never heard that, but that'd be great for the drug company to say, hey, this thing that we thought was inactive, it actually does something beneficial. That's a good question. All right, one more time. Remember when amino acids come together, wait a minute, hang on. Here we go. When amino acids come together, the way they make a polymer is an enzyme will take an OH off of the acid group and an H off of the amino group and take away water. It'll take away water. That'll mean carbon and nitrogen want to form a bond with each other. And what's the special name for a bond between amino acids? What's that called? Special bond between amino acids. It's a carbon-nitrogen bond. Again, I haven't seen you since last week, but this is what you guys are going to have to go back over and look at this. It's called a peptide bond. So if you put a lot of amino acids together, you get a polypeptide chain. Now, it's not a real protein yet because it has to fold into its shape before you can call it a protein. You get that? So there's not a whole lot of difference except it hadn't been folded yet. Okay, now let's go to the next part. What's the next one? I'm not going to spend very much time on this because the book doesn't even mention it. But the second kind of organic molecule that's important to living things, they're called lipids. Now, they're not polymers, and what you have is you have carbon, hydrogen, oxygen. but you have an extremely high ratio of H2O. Have you heard of things like cholesterol, lipids? They have triglycerides. If you go get your blood tested, they test for lipids. They want to know how much lipids in your blood. Okay, so this one I'm not going to spend a whole lot of time on. I will tell you it's made of carbon, hydrogen, oxygen with a very high ratio of H2O. H2O. There. I forgot to say one other thing. Sorry. The names of proteins usually end in "in." That's one ending, like insulin, penicillin.
Oh.
Okay, now if the protein is an enzyme, I bet the biology people know this.
It's an ACE,
What?
right?
ASE,
Like lactase.
right? So lactase might be an enzyme that knows how to break down lactose. Or you guys had DNA replicase or helicase. Helicase is an enzyme that can unzip the DNA helix. And so there's all kinds of, so enzymes end in "in" or "ace." Okay, let's jump down now. Let's jump down to carbohydrates. I'm going to do carbohydrates next, all right? I'm going to give you some advice. If you know you're going to go home and you're going to read all these pages carefully, then I get it, okay? But I'm going to try to give you a good framework to look at. And then when you go back and read it, then you can understand a little bit better. Carbohydrates are very important to living things, and carbohydrates are polymers. Some of them are, and the monomers are actually called sugars. And so we have different kinds of sugars. All right, I will tell you that carbohydrates have carbon, hydrogen, oxygen in them, and they have a 2-to-1 ratio of H to O. So the most famous one is probably glucose. Notice a 2-to-1 ratio. Here's table sugar, which almost 2-to-1, very close to 2-to-1. Anyway, it's like that. I do remember being in my biology class 45 years ago in college, okay? I'd never had any organic chemistry. I didn't know anything about model building. I had no idea. And they would show me these pictures like, I'll show you a picture they showed me. It was looking like this. It looked like a hexagon attached to a hexagon, another hexagon, and it said GLU, 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 and it kept going on. I had no idea what they were talking about. And does anybody know what this is?
Glucose.
It's glucose. And so what they did is they drew the stick structure. I didn't know what a stick structure was. They would draw the stick structure of glucose and attach it to another one, and it was a polymer. And so anyway, if you take a look, if you happen to have your books with you right now, let me show you. Okay, here's one. All right, take a look at this picture here. Whoops, let's see. Where am I at? Here we go. Okay, take a look at this right here. These, how do I know that these are sugars? And in this case, these are both C6. Everybody on here is C6H12O6, but there are different ways you can arrange them. I didn't know what isomers. Nobody ever told that word to me before, but I happen to know that there were 3 sugars that had the same formula, and glucose, galactose, and fructose. Have you guys ever heard those before in any of your classes?
Maybe in like food.
And a sugar is going to end in OSE. OSE. All right, so what would make a sugar want to join to another sugar?
Sweet.
And what you do is an enzyme would take an OH off of one sugar, an H off another, and now instead of carbon-nitrogen bond, it's a carbon-carbon bond, and then you can put a, fancy word here, be careful, a monosaccharide can be attached to another monosaccharide to make a disaccharide. Saccharide means sugar. What if you had 3 sugars, you put them all together, it'd be a trisaccharide. But what if you had 2,000 sugars? Polysaccharide. So here's what you know from your house. Here's what you should know by being a regular human. Ready? If you want to go home and find simple sugars, get some Karo syrup. The Karo syrup is a monosaccharide. Everybody get that? If you go to the hospital and you can't eat, but they know they need to feed you. They're going to put a bag, and they're going to put a PICC line here, and they're going to feed you with that bag. And that bag is actually glucose, but it's a type of isomer. They call it dextrose. So if you go to the hospital and they're going to give you dextrose, that's a simple sugar, C6H12O6. And they're going to feed you that way. Got that? Okay. So what if you put two sugars together? What is the sugar at home that I get in the sugar container? That's called sucrose. And sucrose, if you'll take a look right here, sucrose is, glucose plus fructose put together. And it's called a disaccharide. So at home, Kaware syrup would be your monosaccharide, but the table sugar is your disaccharide called sucrose. And then if you go stuff a piece of bread in your mouth, starch is a polysaccharide. So starches are polysaccharides, long chains. So here's something kind of interesting. What would the formula be of a disaccharide made with two glucoses together? What would the formula be? So let's do some fancy math. Let's see, I have C6H12O6 plus C6H12O6, but they aren't going to go together unless I do what? What do I have to take away? Say it again. Okay, so I'm going to take an H off of one and OH off another. So watch. This is kind of weird math. What do you think the final formula would be for a disaccharide?
H22.
Say it again.
11.
Wait, say it again.
C12H22O11, maybe?
That's it.
Very good.
And that's how you can remember the formula for sucrose, a disaccharide. Here's what I might ask on the test. What would be the formula of a 5 sugars put together in a polysaccharide? So 5 sugars would be 30 carbons, 60 hydrogens, and 30 oxygens, right? But how many waters would you take away to join 5? If you want to join 5 sugars together, you take away 1 water, 2 waters, 3 waters, what? How many waters do you take away to join 5 sugars? 4. What if you want to join 100 sugars? 99. Yes. Anyway, it's kind of weird math, but if you want to know the final formula of a 5 sugar compound, you could do it this way, and there it is right there. Kind of neat. All right, so I want to show you, again, I had no idea what they were doing here when I saw this in college, because I didn't know what a stick structure was. But does everybody understand that these are carbons where the sticks come together? And then the other things are sticking out, right? And so here is a polysaccharide. See what they're doing? Now, I do remember when I had to take a test, I had to remember this. And again, I didn't know enough about it, so I had to do memory tricks. What if you have a long chain of amino acids? A long chain of amino acids is a polypeptide, is that right? And a polypeptide will turn into a protein. But what about a long chain of sugars? A polysaccharide, that would be a sugar or a starch. So again, if you were to eat starch, you're eating a bunch of sugars. If somebody says, I'm not going to eat any more sugar, okay? I'm just going to eat biscuits. I'm going to eat bread. Well, that's not going to work because bread is sugar. Bread is a long chain of sugars. When you put it in your body, the enzyme is going to chop them up into disaccharides and then monosaccharides, and you are eating sugar. Okay. One thing I thought was interesting.
You are sugar.
One thing I thought was very interesting about this is that in the living world, the biological world, and I'm not your biology teacher, but one type of polysaccharide that you may hear of, and let me see if I can get my handout sheets. Shoot, where's my handout sheet? Here it is. Let me go back to the original handout sheet that I gave you. Here it is. Okay. All right, so what this person did, they actually typed out some of this stuff. So here's a monosaccharide here, and here's a disaccharide put together here. And then glucose and glucose makes maltose, and glucose and fructose makes sucrose, like that. But I want to show you something here. What about a polysaccharide? Now, do you know that, have you ever taken a plant out, pulled it out of the ground, and you ever broken the root? You say, well, no, I don't go outside. I like to look at my phone a lot. But no, you could also say, have you ever been outside and you pulled out a plant and there was a root and you were just interested and you actually broke the root and you saw this milky stuff? Has anybody ever seen that?
It's like when I was like younger.
That's what I
Okay.
had for breakfast.
Okay, but anyway, so what
That's
you're
what I
looking
had
at,
for breakfast.
what you're looking at is starch.
Oh.
And so what happens is the plant makes all kinds of sugars by photosynthesis, and then it puts them together, sends them down to the root, and stores it as a form of starch. Now this is plant starch, and we call it starch. Okay, here we go. Now animals do the same thing, except they have a fancy name for, the fancy name for plant starch is starch. And when you buy starch in the store, it came from a plant. But they have a fancy name for what your human body does. The human body also takes sugars and stores them in a form of animal starch. Have you ever heard that word before?
Like,
Glycogen. And when you're not eating, your body's probably using glycogen, and it's actually saying, give me some more of that glycogen, I'll break it down for my sugars because you haven't eaten for a while. And so a fancy name for animal starch is glycogen. All right, another thing is this paper. This paper is made of another polysaccharide called cellulose. Now, cellulose is what cell walls are made out of, and 2x4s, and plants can also put sugars together to make long chains there. Now, here's the only thing that's different. And again, if I was a biology teacher, I'd show this to you, but imagine sugar in a straight line. That might be animal starch. But I might do, cellulose might be sugar, branch, sugar, branch, and it might have branching sugar chains, and that might be like cellulose. Yes?
what kind of tree do they make paper from?
Even though I'm not in the paper business, I know pine trees are big and they grow fast. So a lot of companies will plant lots and lots of pine trees. They grow very fast and they harvest them. They don't harvest all of them. They harvest a certain amount. They go to another area. They plant some more. But most of the time, I think it's pine trees, but I'm not certain about that. But almost all your trees are going to make cellulose. That's the cell walls. That's the wood part.
And that's what they make the paper out of?
So they grind it up, they make a mulch out of it, they use some chemicals, they make it into a old-fashioned paper. Have you ever seen do old-fashioned paper out of real wood? Okay, and they actually squeeze it out, they squeeze the water out into a screen, and they let it dry, and it's kind of like paper.
You've seen it TikTok.
You probably went to some field trip.
Probably have on TikTok.
I know you have.
Okay, let me real quickly. I want to show you. I forgot to mention this a little bit. Lipids. One thing about lipids, and that I forgot to mention here. Besides having a really high hydrogen to oxygen ratio, they. And I remember the first time I heard this word, I giggled a little bit, but not too loud. But lipids are made of glycerol and fatty acids. Okay. Thank you. But anyway, fatty acids are these. Look at this right here. Hey, everybody, look at it. Wait a minute. Look. I taught this to you before. What happens when you only have carbon and hydrogen? Is it going to be polar or non-polar?
Polar. Non-polar.
Right. Gasoline is only made of carbon and hydrogen. It's non-polar. Okay. Now, what's interesting about a fatty acid is it has the fatty part, which is non-polar, and at the end it has C double bond O OH. It should have OH here. So we have an acid part which is polar. It should be C double bond O OH. You didn't write that right. So what happens is, if you remember a biology lesson from a long, long time ago, lipids help make up cell membranes because
Jellyfish.
see these little balloons? I didn't know what those little balloons meant when I got to college. And what it was showing is, here's wait, I'm gonna stop.
Stop collaborating.
This was this thing called glycerol, and here's a fatty acid, and here's another one. And what happened is one end of the lipid, it liked water. The end that was acid, it was hydrophilic. I like water. And the other end was like this, and it was hydrophobic because it was nonpolar. And so we now know that the cell membrane is a double layer of this, made of lipids with proteins embedded in it. And this end likes water. And where do you find water? On the outside of the cell and on the inside of the cell. But what part doesn't like water? And that's right here. So when you're looking at a cell in a microscope, you probably remember seeing a cell that looked like this. Remember the cell membrane, the nucleus? With an electron microscope, you would see that that line is not one line, it's actually two lines. And that's what this was all about. And so if you were to take college and they start talking about this kind of stuff, maybe that'll come to you. All right, the last thing is kind of the fun part. The last polymer that I do want to talk about, by the way, lipids are not polymers, but the last one I want to talk about, and I do need you to pay attention on this one, this has to do with the fourth type. What's the fourth type of organic molecule that's important in living things? We did proteins, we did lipids, we did carbohydrates, and the last one's called nucleic acids. Now again, I have no idea where you stand with your belief in God. I don't know where you stand with any of this beliefs of anything. But I'll mention something to you here, okay? Here we have a situation where every living thing we know on this planet, it seems to have this in it. It has nucleic acid in it. Now, what's its purpose? Here, this is for structures and for fuels. Structure and fuel. Mainly fuel and structures, but not this one. Nucleic acids are not used as a fuel, and so, hang on. It seems like the purpose of this thing is information. I think that's very, very unusual. So living things have chromosomes or DNA material. That's made of nucleic acid wrapped around protein. So the nucleic acid has information on it. Why would you need to have information? And that information is the information that can make this cell be able to pass on the information to make another cell just like it, et cetera, et cetera, et cetera. And so its only real purpose is information. So what's interesting about it is, well, how is it coded? How is the information coded? Now, you guys live in a world, a digital world, and everything you can think of, every movie, every song, is actually a series of ones and zeros. 0, 0, 0, 1, 1, 0. That's how loud it should be. 1, 1, 1, 0, 0. That's a note. Everything in your whole world is 2 letters in the alphabet, and you get all that stuff. It's all stored with an alphabet that only has 2 letters.
Wait, what letters?
Zeros and ones.
Well, I get, so is that O and L?
But 0 and 1, it's like on and off, really. It's like on and off. Okay. Anyway,
42.
hang on.
The
Hang on.
That's the secret to the answer.
Wait.
What's the question? But the Earth was made.
All right, hang on. I'm trying to get to that other part of your brain. Okay, I'm trying to get the other part of your brain.
The one that you don't.
I want you to think about this. We didn't find out for a long, long time that living things had information molecules in them, and it was stored in a form of 4 letters of the alphabet, and that's the A, T, C, and G.
What about R?
It's not in there.
Okay, no, wait, hang on. We'll do DNA. We'll do DNA first, okay? Let's do DNA first. So let me show you what happened here. If you have this handout sheet, let's talk a little about this. Nucleic acids are polymers, but what are the monomers? Okay, now the monomers are called nucleotides. So I'm going to stay with me, and this is the last part of the chapter.
Wow.
Okay, when I saw this a long, long time ago, I remember seeing this in a biology book, and some of you have seen this too. It looks like a ribbon, doesn't it?
Yeah,
2 ribbons?
SAT.
As a matter of fact, the ribbons not form one helix, but they form a double helix.
Helix.
And about 55 years ago, Watson and Crick won the Nobel Prize for discovering the structure of DNA. Not only was it a double helix, but they discovered that on each strand of the helix there were bases, and the bases went inside. This other guy, Linus Pauling, who had already won a Nobel Prize in chemistry, he was hot on the same question, but he had some evidence in one of his experiments that the bases must stick out because they have to be red. And he went with all his experiments based on that, and he lost this race. Watson and Crick happened to have, they went to a person who was an X-ray crystallographer, and it was a woman.
Wow.
And back then, I'm sorry, but back then it was similar, Rosalind Franklin was not recognized, but all of her X-ray crystallography pictures, it led them to the idea, wait, we had the wrong idea. The bases must be sticking in. Now, later, she was not on the original Nobel Prize. It wasn't until later that they confirmed that without that information, they might have taken the wrong turn, and then she was awarded. I think she might have been dead by then, but she might have been posthumously. So Watson and Crick had a little bit of an edge because they had some evidence that Linus Pauling didn't have. So let me show you how this all comes about. I did not know what this meant when I saw that in a biology book. I remember they talked about double helix, but I didn't know what they were talking about. But this little ribbon, see that? This little ribbon is made of chemicals. And if you're in biology class, here's what you should know. The backbone of this little ribbon is made of sugar and phosphate and sugar and phosphate and sugar and phosphate. And so there should be an S here, another phosphate, S here. And everywhere there's a sugar is going to be attached to one of these bases. And I'm going to try to tell you a story here, okay? Hey, I'm going to stop. Okay, if you want to leave, you can, but I need to finish this, all right? Okay, so I'm going to tell the whole story, but I need you to stay with me on this story. All right. So let's start back here. First of all, they found out that there is this monomer, which is very complicated, and the monomer was made of 3 parts.
Are we still talking about Watson and Crick?
Dude, this is not
Yes,
the time.
yes. Matter of fact, Watson and Crick, they had the basic idea of how it was made, but they actually made models. Instead of cardboard, they used little sheet metal, but they had adenine, a model of adenine cut out as a flat model, and then they had guanine, cytosine, thymine, and they were trying to put them together, but the photographs from Rosalind Franklin showed that the double helix had exactly the same distance between this base and this base as the other ones. And they knew there were 4 bases in there. They just couldn't understand how they could pair together. And so whenever you put adenine and thymine together, it was bigger. It was wider than it should have been. I'm sorry, adenine and guanine, sorry. But when you put adenine and thymine together, the distance that they were bonded together was exactly the same as guanine and cytosine. And that's where they got the idea that there were bases that were paired together. And I'm going to show you that to you in just a minute. Okay, so first of all, you have a sugar, a phosphate, and a base. Let's talk about the sugar first. There were 2 kinds of sugars found in nucleic acids. And I didn't know this for a long, long time. Not in college. First time I heard it was college. If the sugar, look at the sugar here. This has 1, 2, 3, 4. 4 of the carbons are in here, and these are 5 carbon sugars, and one of them's up here, like a little periscope. See a little O there? Okay, see this ring? This is a ring structure with 4 carbons in it and 1 up, and that's called ribose sugar. Now, when they discovered another sugar, it looked identical. It was identical to it, except instead of being OH here, it was just H. This one had already been named. It was called ribose. They called this one Jerry. No, I made that up. They decided. You made that up. Ready? Next. Ready? This was called ribose, and they very thoughtfully said, why don't we simply call this deoxyribose? ribose.
I mean, that makes sense.
And that would be the same thing as ribose, except you're losing one oxygen. So that's when they found out later that DNA always uses a sugar called deoxyribose sugar, and that's why it's called deoxyribonucleic acid. And then for RNA, it always had ribose sugar, and that's why it's called ribonucleic acid. So now we got the sugar part done. Next, the phosphate group is PO4 phosphate. You know what phosphates are, and those are identical for everybody. DNA, RNA, same thing. But this is where it is different. There's a base, and this base, and I'll show them to you in just a second here. The base is found in DNA right here. They found 4 bases in DNA: adenine, thymine, cytosine, and guanine. And they found out that adenine always base pairs with thymine. I'll show you why that works later. And since we live in North Carolina, in Greensboro, we have A&T University. That's a great way to remember that adenine bonds with thymine. And we have UNCG. That's a great way to memorize that guanine always bonds with cytosine and vice versa. Okay, so there are 4 bases in DNA, and they stick right there. And the reason they're called nitrogenous bases is because they have a nitrogen atom in them. Okay, next. When they looked at RNA, instead of being double-stranded, instead of being a double helix, it was only 1 strand. And it had adenine, uracil, cytosine, guanine. And I'll show you a picture in just a second. Uracil is very, very close to thymine in structure. Very close. So in a way, whatever thymine used to base pair with, adenine, what does uracil base pair with? Also adenine. You see that? And I'll try to tell you how this all works, all right? So we already know what the sugars are. Let's go to the next part. Let's go to the book now. All right, here we go. All right, stay with me. Everybody ready? You can do this. All right. What do you call a sugar, a phosphate, and a base?
DNA.
Not yet. It's the building blocks of DNA.
Hey.
It would be called a DNA nucleotide. So the monomer of nucleic acids are called nucleotides. Now this right here happens to be a DNA. I happen to know it's a DNA nucleotide because, see right here? This is deoxyribose sugar. Okay, look what they did here. Imagine this nucleotide and then this nucleotide joining together. Sugar, phosphate, sugar, phosphate, sugar, phosphate, and that's what this ribbon is. Sugar, phosphate, sugar, phosphate. Now where are the bases sticking off? The base is sticking off of the sugar. The base goes in here. So if you take this in college, you're going to hear about DNA being like a ladder. Okay? The rails of the ladder are sugar, phosphate, sugar, phosphate, sugar, phosphate. The rungs of the ladder are the bases. A and T, C and G. Everybody got that?
And they're connected.
Okay, now listen to me. Listen now. Hang on. The sugar-phosphate bond, very strong covalent bonds. They very difficult to break those. But what about the rungs of the ladder? What and Watson and Crick discovered this. They weren't great at chemistry. You know what they did? They went and found some people who were great at chemistry, and they asked them this question: if I were to put adenine and thymine together, why would bond them together? Oh, hydrogen bonding. There are three hydrogen bonds right there, and there are 2 hydrogen bonds in the other one. And so the kind of what holds them together are hydrogen bonding. Now, not as strong as covalent. You say, well, why wouldn't you want to have strong? I'd want these to be strong because I want the sequence to stay the same. C, G, A. See that? I don't want the DNA sequence. That's called your genetic code, right? I don't want the genetic code to change. But what do I want to do if I had DNA? Why would I want this to be not super strong here? Yes?
So you could break it in 2. So
Right.
RNA
So it can unzip, it can come apart, and this half can make a new piece of DNA, and this half can make a new piece of DNA, and where I used to have 1 piece of DNA, now I have 2. And so for DNA replication to take place, you have to be able to unzip. That was another part of Watson and Crick's discovery. Yes?
Aren't we able already to
No.
tamper with the genetics? and make
Yes.
bonds?
CRISPR,
Like with,
There's some scary,
Like
yes.
Cas-
I think there's some scary, okay, wait a minute. I think there's some, think there's some scary things out there. CRISPR, if you want to read about CRISPR, we have the ability to actually build pieces of DNA with the sequence that we want. And that's very scary to me.
Is it
Uh,
also
improving
helpful because it
people,
can cure cancer?
Yes, and that's the hope, is that you could use it for good. If we weren't sinful, if we had no evil in us, then I would not be worried. But I promise you, there are people that are using CRISPR technology, and they would love to make a weapon.
To destroy gene editing.
Right, and if they can make a genetic, if they can make a certain kind of a piece of DNA or RNA, and they put it in a virus, if they wanted to, they could make it so specific that it would kill a certain type of person. So they'd say, I'm not going to kill this kind of person, but I'll kill this kind of person. And I wish that we didn't have that evilness in us. And please don't go home and think, oh, I don't have any. Everybody has that. All right, so let me show you. Here's a picture I was trying to show you. Hey, talk about later, okay?
This is relevant.
So here's your ladder. Okay, wait a minute. Here's your ladder, and you have sugar phosphate, and here are your bases in here, and this is hydrogen bonding right here. Now I want to show you, they actually did cut these out of tin metal, and they took us to a chemist, and they had a picture of, let's see, I'm sorry. Here, I'm on the wrong page.
Me too.
Oops. Hey there. Okay, now, can you take a look at this right here real carefully? Now, be a science person, okay? Here we go.
Parker, you're a science person.
This right here is guanine, thymine, adenine, and guanine. I'm sorry, this is cytosine. Notice that these have just one ring structure, and these have two ring structures, okay?
Well, they can.
And if you're taking biology class, your teacher may have said that some of these are called pyrimidines and purines. Did you guys use that word at all?
Yeah.
Okay, so it's either the double ring structure or the single ring structure, right? Now, look at thymine and uracil. What's same?
Different.
And the only thing different is in having just an H here, you have a CH3. That allows uracil to act kind of like thymine, but what's interesting, and I think it's very interesting, you never find uracil in DNA. You always find thymine. And you never find thymine in RNA. You always find uracil. And I think that's kind of interesting. So now we have, God said, I want to have a certain code that's called the genetic code with DNA, but I have to transcribe that into another message. And it's going to be a message which is similar, but not identical. As a matter of fact, it's not going to have any thymine in it. Okay, so stay with me a little bit. Let me show you, let me see if you understand how this works, okay? All right, everybody take a look, and does everybody know that if the base over here on DNA is G, then what's going to be on the other side?
C.
Okay, and T and A, and so we already know that that's the double helix, that's DNA. Okay, so here's the story, and we're almost done. One of these strands of DNA will come open, and the enzymes will start to make, instead of using deoxyribose sugar, they'll use ribose sugar. And so they're going to make not DNA, they're going to make RNA. So here, look right here. Here's an RNA nucleotide. Here's the base sugar phosphate. Now, here's how it goes. If DNA says AAT, then the RNA is going to be UUA. Okay, because adenine and thymine, but there's no thymine in RNA. Get that? So here's another thing that Watson Crick did, which I thought was amazing.
Throw that on.
Okay, hang on. A few more minutes, yes.
So why does T need a replacement, but C or G doesn't need a replacement?
I don't have the answer to that. That's a great.
We're gonna have to take it up.
I mean, the fact that we discovered it, I don't know exactly why. Here's what I thought was very ingenious with Watson and Crick. Okay, they said, here's what they said. They said, okay, so we have evidence that DNA. Okay, we have evidence that DNA makes a type of RNA that's going to leave the nucleus and go out into the cytoplasm and make proteins. at the ribosome. Now, look right here. We didn't know, how do you translate the message? And they said, all right, maybe one base means something. When the ribosome reads U, maybe it says something. But if that's the case, then how many different amino acids could you call for? U says, go get this amino acid. What would C be? Go get a different one. And G, and how many can you call for? You can only call for 4 amino acids. That didn't make sense then, because how many amino acids had they found? 20.
20.
There are 20 different amino acids. So here's what they did. Ready? They said, okay, we think the message is read 2 bases at a time. Now, how many messages can you make there? Let's draw them out. I can have UA, U what? UG, UC,
No.
UU. Is that right? And what else can we have?
AU.
Okay. All right. We can have AU. So how many could we make if we read it 2 at a time?
4 factorial.
All right.
4 factorial.
Okay. So, all right. How many?
Pretty sure it's 4
It-
factorial.
4 times 3 times 2.
Okay. So we have 4 bases. If they're read 1 base at a time, you can only call for 4 amino acids. If you read them 2 bases at a time, you could call for 16 amino acids. But that bothered them because they knew there were 20 amino acids. How could you call for 20 when you only have 16 different messages? So they decided, and they took a chance here. They said, I think the bases are read I think the RNA is read 3 bases at a time, and they made, this is called a codon. A codon is 3 bases in a row on RNA, and a codon means something to a cell. Now, if I was in biology class, I'd give you this nice little chart. Do you guys have a little chart?
Yes, we
And
did.
it's a decoder. And it would say UUA. UUA says, go get this amino acid. UUC might be a different amino acid or the same. But how many could you call for? 64.
60.
Now, we don't have 64 amino acids, so there are duplicates. Wait, listen. This is kind of neat part.
Look here.
So if UUA calls for the same thing as UUU, it's possible that if you made a mistake, like a mutation, you still be okay. Because you might have UUA, UUU might call for the same one. But if you make the wrong mutation, it's going to call for the wrong amino acid, make the wrong protein, everything's bad. So I thought it was kind of neat that Watson and Crick figured out that the RNA is read 3 bases at a time. This is called a codon. And the last part of the story,
That's their last name.
well, Watson, Crick was an English scientist, and Watson was an American scientist, all right? And they're still, I think they're both still alive today, but I will say that Crick has gotten into some weird things, so if you want to look him up, you might, some people think he's kind of,
Gone crazy.
I don't know. I don't know about crazy, but he's,
Senile?
not necessarily, he has some weird ideas. He has some different ideas. No, I don't know if he's left-handed or not. Okay, last thing, here we go. Last thing, here we go. It's amazing. So the DNA has a genetic code. The DNA has to make a messenger RNA. The message goes out of the cytoplasm, finds the ribosome, and says, read me 3 bases at a time. That's a codon. And that's going to tell you what amino acid goes first, and second, and third, and fourth, and fifth. Okay? Now, if you're in biology, you probably got way more complex than this. You know there's a start codon. There's even 3 bases that mean ignore, 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 start. There's a lot of bases that ignore, 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 start, stop.
Don't we only use like a little bit of the code? Like
Right, and even though for a while people thought that all that DNA that didn't seem to code for anything, they said it's just trash. They have some second thoughts about that now. They're thinking that it actually is a way of having variety, eventually have a variety. Okay, last thing about the story. Ready?
Oh.
The RNA has been read and translated and says, hey, you'll get this amino acid. But how does the amino acid know?
Test tube baby.
How does the amino acid know to stop at that exact codon? So watch right here. If the messenger RNA had CC, Wait a little bit longer, okay? If the messenger RNA has CCC, that does code for a certain amino acid.
CCTV.
But who brings that to them?
Messenger RNA.
No, but you're close. It's transfer RNA.
Transfer RNA.
Now, here's what's interesting about transfer RNA. RNAs are single-stranded. Look at this drawing I have here.
Is that a
Notice,
cross?
look at it. See that little thing here? It's single-stranded, but some parts of the strand are attracting other parts of the strand into little loops and things. And it looks like that. So here's the last part of the story. Last part of the story. How does the transfer RNA, who's bringing the right amino acid, how does it know to stop here? And so if this is CCC, there are 3 bases that stick off of the transfer RNA, and this would be G, G, G. And if this is a codon, this is called the anticodon. And so what you have is the codon is the message, and the anticodon makes sure to stop in the right place. And it's fascinating. If you have a chance, and I hope you do read this, if you read this before Wednesday, I think it'll all come together for you, all right? If you come to class on Wednesday and you haven't read some of this, you're going to have a lot of questions. Say, I don't know the answer to that. I know the answer to that.
Where is the codon located? Is it on the
Okay,
protein? Is it
so the codon, okay, the codon is 3 bases in a row on the messenger RNA.
Okay, got it.
Oh, it's on the messenger RNA?
But the anticodon's on the transfer RNA, and that's how it knows how to stop at the right codon, like that. Anyway, if you, I don't want to necessarily want to show you, I don't have time to show you a little video, but I have a video, there's a video online where somebody made an animation
Hey, Bob.
of this in real time.
It's really hard.
In real time.
Should we watch it?
And it's amazing. I only have 5 minutes, but let me see.
5:10.
Let's watch it. We got time.
Go.
Wait, is that mine?
What was that one? Did you hear that?
That was Jeffrey.
Ew.
Oh my god.
It's in your hair.
It's not. I heard it.